Welcome to the first screencast on biodiversity. This is one of two screencasts. This particular one looks at the idea of biodiversity and also mechanisms or ways in which we can sample organisms in ecosystems. Okay, so the first slide here starts off with some definitions, and the first definition is that of species. Okay, and I think we've defined this before. It's a group of individual organisms, very similar in appearance, anatomy, physiology, biochemistry and genetics, whose members are able to interbreed freely to produce fertile offspring. Okay, the obvious example of this is the horse and the donkey. We can define these as coming from different species, even though they can interbreed, okay, because the products of them interbreeding are mules, okay, and mules can't interbreed and therefore infertile, okay, so that's the business about being able to produce a viable offspring or fertile offspring, okay, so that would mean that horse and donkey would be of separate species, okay. So moving on to the next definition, we have habitat, and this is simply where an organism lives. Okay, there can be all sorts of description of habitat, and it's a description of the abiotic and biotic factors that exist in a particular region. Okay, so here we have types of habitat. So we could have a grassland habitat, deserts, freshwater marshes, tundra, a land cave, tropical rainforests, scrub or chaparral, temperate deciduous forest, and temperate ponds. Okay, would all be examples of types of habitat. But even within habitats, you can get microhabitats. Okay, so certain organisms, like a woodlouse, might live under a stone. Okay, so that might be a microhabitat with its particular amount of light and dampness and temperature, etc. A description of a microhabitat. Okay, so that's what a habitat is, essentially where a description of where a particular species might live. Okay, the next word that you need to be able to define is biodiversity. Okay, and this is a slightly more complicated word because it's got three components to it. Okay, biodiversity would be all the different plants, animals, fungi and microorganisms, all the species of all of those worldwide. Okay, it would also look at the genes that or alleles that each of those species contain, and also the ecosystems which they form part, or the habitats they live in. Okay, so we've got essentially the idea of species diversity, all the different species, irrespective of what kingdoms or domains they come from. Okay, we could then look at the alleles in each of those species, genetic biodiversity, and then lastly habitat biodiversity. Okay, so the idea of biodiversity, three components. Okay, species, the genes within a species, and the habitats in which the species live. Okay, that is further explored on the next slide. Okay, so this, as I said, this slide here, it says explain how biodiversity may be considered at different levels: habitat, species and genetic. Okay, so looking at each of these, so habitat biodiversity, okay, this is a description of the different habitats that can be found in an area. Okay, e.g. a deciduous woodland provides a large range of habitats for a large range of organisms, whereas a coniferous woodland provides fewer habitats, therefore supports fewer species. Okay, so we would say coniferous woodland has a lower habitat biodiversity than a deciduous woodland. Okay, moving on, we then have species diversity. Okay, and this is the variety and abundance of different species which inhabit an area, and we will move on to look at measures of species diversity on the next screencast. Okay, and lastly, genetic diversity. This refers to the variation of alleles for a particular gene found within a species. Okay, So genetic biodiversity is of particular concern when conserving very small numbers of large mammals and zoos, etc. And what we find is the low population leads to low genetic biodiversity, which can lead to all sorts of problems. So conservationists and zoos have to be quite careful to try and what we call outbreed, to try and increase the genetic biodiversity amongst individuals. Okay, so genetic biodiversity is the variation of alleles within a particular population of a species. Okay, representing all that diagrammatically. Here we have genetic diversity in this vole population. So this is looking at the alleles that are contained on their genes. Okay, on their chromosomes here, the different alleles that might exist. Okay. This looks at the species diversity within a particular area. So we look at all the different species, the plants, the trees, the fungi, the amphibians, mollusks, and the mammals themselves. Okay, so it's the different species, but it's also the abundance of each species which is of interest. And as I say, we'll come back and look at how we measure not only species diversity but the abundance of individuals within each species. Okay, and then lastly we've got habitat biodiversity. Okay, that's just called ecosystem diversity here, but from our nomenclature we call it habitat biodiversity, which is all the different habitats that might exist within a particular region. Okay, and I think my example here, we have what looks like coniferous woodland here on this side of the river, okay, which is relatively limited habitat biodiversity, whereas over here we've got deciduous woodland, okay, with bushes, etc., which has a wider range of habitats within here. So we'd expect to find higher species diversity in here because of the higher habitat biodiversity. Okay, so just in summary, so you need to know that this biodiversity can be considered at three levels: habitat, species and genetic. Genetic refers to the variation of alleles within a particular population of a species. Species diversity talks about all the different species and the abundance of those species within a particular area. And habitat biodiversity looks at all the different habitats that exist in a defined area. Okay, so remember biodiversity: habitat biodiversity, species diversity, and genetic biodiversity. The third slide here looks at the idea of sampling and gives examples of how we go about sampling. Okay, so when investigating species diversity, we need to know all the species present and the abundance of each species in a particular area. Okay, now this is not really practicable because of the size of potential habitat and the amount of counting that would be required to work out the entire population of a particular species, or even all the different species that might exist in a particular habitat. So what we do is we take a sample, and that sample is hopefully representative of what occurs in the whole population. And on the next slide we'll talk about how we try to ensure that it is representative. Okay, but this slide just actually looks at the ways in which we can sample. And so we're sampling, we're studying a small portion of a habitat. Okay, so and if you want to estimate a population, then we multiply up the area of that we've sampled with the whole area of the habitat. I'm sure you remember estimating the number of earthworms on the water meadows in the lower school, where we used a quadrat to count the number of earthworms in a particular area, and then we multiplied that up by the area of the whole water meadows to get a very large number that we estimated as the number of earthworms in the water meadows. Okay, so that was an example. Another example might be if you're trying to estimate the number of aphids on a bush. You could count the number of aphids on one leaf, and then estimate the number of leaves by counting the number of leaves on one branch, and then counting the number of branches, and then multiplying up that by your number of aphids to give you an estimate of the population of aphids on the whole of the bush. Okay, so those would be two examples of sampling. Okay, now we're just going to look at various bits of apparatus that ecologists might use to sample, and there are key words here like quadrat, point quadrat, transect. These are words that you should understand what those are. Okay, and I'll quickly go through those one by one. So a quadrat, which you will have used before and you'll have the opportunity to use again, is simply a square frame, usually ½ metre by ½ metre. Okay, and some of these quadrats are divided into even smaller squares, so it can help us count numbers of things within that quadrat. Okay, that quadrat will be placed in our sample site. Okay, and that will give us, when we can count within this quadrat, the number of particular species, or the different species, and record different species and the number of each different species that occur within that quadrat. Okay, the other idea, we can use these quadrats in things called transects. So here's a transect where we've put a line randomly through some habitat, and what we do is that we place our quadrat at points along this transect line, and that can give us a random way of counting the number of different species and getting a representation of what exists in the whole area. Okay, transects can also be useful in finding the effect of one variable on the amount of plants that exist. Okay, so if you when we talk about succession in the upper six, the idea of putting a line or a transect from the strand line on the beach back through the dunes right to the climax community, and then at particular at individual distances along our transect line, what we do is we place our quadrat, and then we can work out the number of species there, and we move, let's say, every 10 metres up the beach until we reach the climax community. We will get an indication of the change of species as we move away from the strand line, and that's how we can study succession, which is something you'll study later in the course. So that deals with, actually I haven't dealt with point transects. Here's a device which helps us look at small plants, okay, or can also be useful for when we've got layers of plants. So we've got lots and lots of plants on top of each other, and what we do is we put one of these frames, which has got a knitting needle type device, which we place through a hole. What we do is we record every species that the knitting needle touches. Okay, so that's called a point quadrat. So we place that point quadrat, we could say place this point quadrat on this transect line over here at certain distances and take readings through this transect, okay, using a point quadrat rather than this square quadrat that we've got here. Okay, now moving to animals now. Before we go to animals, some plants are very difficult to count individually, grasses for example. So what we can do is we can take quantitative values such as percentage cover, or there's a ACK4 scale as well, okay, which is just a qualitative judgment that ecologists will make on the abundance of a particular species in a quadrat. Okay, A standing for abundant, C standing for common, F standing for frequent, O standing for occasional, and R standing for rare. Okay, so the individual just takes a judgment on and gives it a qualitative label according to the judgment that he or she has made on the amount of a particular species in a quadrat. Okay, quantitatively we can work out percentage cover. this quadrat down here illustrates this very nicely. So I've got a quadrat here. Each square is worth 4% of the total quadrat here. So this species A, we can count up 1, 2, 3, 4, 5, and roughly takes up 5 of our squares. So each square is 4%, so we can estimate that species A has 20% ground cover. Okay, that's a more quantitative value that we can take. Moving on to animals. The problem with animals clearly is they move around, and we need to have ways of sampling those, and I'll go through some of the techniques we can have. So sweep netting, pouters, pitfall traps, Talgren funnels, a light trap, kick sample, mark capture, recapture. I'll just quickly go through all of those very quickly. So here's sweep netting. Okay, so you've just got a net that we're sweeping through foliage, catching flying insects, and we'd have to have a system of randomly moving through what the sample area with our sweep net. Okay, so that's sweep netting. Similar approach, but in water. There's a kick sample. This is looking at sampling invertebrates in a riverbed. So what we do is that we get our net, and we then kick and disturb the riverbed, which disturbs all the invertebrates, and the flow of the river forces the invertebrates into our net. Okay, so that would be a kick sample. Other ways, Talgren funnel here. Talgren funnel. This is for estimating insects in soil or leaf litter. And we take our sample of soil or leaf litter, place it in the funnel, which has got a sieve here, put a light on the top. Those insects will move away from the light, fall through the sieve, and fall into this preservative alcohol. So we can then count the different species, the number of each species that have fallen through into the alcohol. We've got other ones here. We've got a pitfall trap and a pooter. So here's a pitfall trap, which does as it suggests. Okay, it's an open pit, okay, which the organisms can't crawl out of. Okay, and you allow this a certain amount of time for organisms to fall into your pit. Okay, and then you can count the organisms that have fallen in there. And another example is a thing called a pooter. You might not have come across before, so I'll just write that word there, pooter. And a pooter is simply a jar with some sort of suction device, so you can create a partial vacuum in here, and we suck insects off leaves of things into the jar here, and we can then count the number of individuals and distinguish all the different species of insects, let's say, on some vegetation. Okay, and then lastly, and probably the more complicated one, is the mark capture recapture one, illustrated by this diagram here. So what we do, so we've got a population of like mice. Okay, so what we do is we're trying to work out the whole population of these mice, and what we do is we capture some mice. Okay, and then what we do is that we release those mice back into the population. Okay, so we've captured them and then marked them. So we've got little yellow splodges we put on their heads. Okay, so we can recognise the ones once we've caught them again. Okay, we then release them back into the population. Okay, and the idea is that these mice here will distribute evenly throughout the population. Okay, which it looks as if they haven't there, but that was the idea, that these mice will then move around and distribute themselves evenly within the population. We then take a second sample at some point later. Okay, so we captured some mice, and what we do is we count the number of mice that weren't marked in that second sample, and also the number of mice that were marked. Okay, and what we can do is that there is a formula we can use to work out the amount of mice or to estimate the number of mice in the population, and that formula to the population would be equal to n1, which is the amount of mice that we've captured first of all, okay, times the number of animals captured in the second visit, okay, times n2, okay, and divide that by the number of animals that were marked on the second visit, okay, which in this thing is n3, okay, so these animals there, okay, so that will give us an estimate of the population, okay. There are various assumptions we make with this, okay. First of all, these animals that we captured all have to distribute themselves evenly throughout the population, so we've got to give them time to do that. And secondly, that the marking doesn't in any way affect the survival of these animals. So possibly this yellow splodge on their head could have attracted these to birds of prey, and there might be less of them, okay, in the population when we do the sampling of N2 than there were when we released them, okay. So the assumption is that that isn't the case, okay. But anyway, that gives us an idea of how to work out population. I don't think you need to know the details of this, although you might be given a little interpretation type question on that, but you wouldn't have to know this formula per se. Okay, so that's quite a long slide which covers quite a few ideas, but it's all to do with sampling. Okay, so it looks at why we sample, because the whole population, it would be impracticable to calculate populations in the whole population, or the number of different species in a particular area, so we use sampling. And it talks us through a few ways of sampling plants, and it also talks us through a few ways of sampling animals. Moving to the next slide. So this slide looks at things that we should consider when we are sampling, and explains why. Okay, so the first thing is, when we sample, we must randomly sample. Okay, the key thing that we want from our sample is that it is representative of the whole population. Okay, so we need to randomly place quadrats, or randomly place R quadrats, or randomly sweep net, or randomly kick sample. Whatever we're doing, we need to do it randomly. Okay, when trying to measure species diversity. So random is a key word that you have to be able to use and trot out in exam questions, and the reason we do that is to avoid bias in your sampling. Okay, so imagine your natural bias. If you had a quadrat and you were trying to estimate the daisy population on a lawn, you might be drawn to parts of the lawn which had more daisies than others, and obviously that would therefore give you that would be showing bias. And the problem with that is that if you then multiply that up, your estimate of daisy population would be much higher than actually existed on your lawn. Okay, so by randomly positioning your quadrat, okay, you have a better chance of avoiding bias, and you would get a more realistic value which would be representative of the whole population. Okay, so bias sampling would lead to data that was unrepresentative of the norm. Random sampling can be achieved by taking samples at regular intervals. So our transect that we used before is if we lay a transect line across our particular area and use and sample at various points along that transect line, and that would be an example of random sampling. Okay, we'd obviously have to randomly place our transect would be a key thing. The other thing is to divide habitats into grids, okay, and give each grid a coordinate, and then use a random number generator to determine which grid would become your sample site. Okay, so just illustrating that. So let's say I have a football pitch I was trying to sample. I would then basically have our my y coordinates and my x coordinates. Okay, so let's say this is a corner flag, and here's the try line and the touch line. And what we could do is that we could use random coordinates. Okay, so we could get, let's say, 5 and 4. So I'd move 5 meters along the try line, and then 4 meters up the touch line, and I would place my quadrat there. Okay, so I've randomly chosen where I'm going to place my quadrat to work out my daisy population, whatever it is I was trying to work out. Okay, so that's using coordinates to randomly place a quadrat or a point quadrat or whatever it was you were using. So your options are to use transects or to use coordinates to split up your area to randomly choose where you put your quadrat. Okay, so that's random is a key word, avoiding bias is a key word, and the other thing that you need to do is to try and get as data which is reliable as possible. Okay, so the more samples you take of your population, the more representative of the situation in your whole population your data is likely to be, and therefore that is going to be more reliable. Okay, so a number of samples should be taken, and then you can take means and averages can be calculated, and the more samples taken, the closer the mean will be to the real value for a population. Okay, i.e. is more reliable. So more samples equals more reliability. Okay, the obviously thing that limits this is the time that you have available for your study would be a practical consideration you'd have to take into account when planning an investigation. So that finishes our look at some definitions of habitat, species and biodiversity. It also looks at different levels of biodiversity, genetic, habitat and species biodiversity, and it looks at sampling, different mechanisms, first of all why we sample and different mechanisms of sampling, both plants and animals, and then the last slide here looks at the importance of random sampling, okay, and looks at why we randomly sample, i.e. to avoid bias, and looks the reason why it's important to take multiple samples of a population so you get reliable data. Okay.